All right, hopefully you're seeing my slides. So first up, we have Amy from the North Carolina DMF talking about the work that they're doing.
Can everybody hear me?
I can.
Excellent, fantastic. Hi, everyone. My name is Amy Staples. I am the tagging biologist for the North Carolina Division of Marine Fisheries Multi-Species Tagging Program. Just to give you all a little bit of update, we are working very diligently to get our current and historical array data into the Matos network. Right now, our current array is the Albemarle Sound Management Area, which includes the Roanoke River, Albemarle Sound, and the Outer Banks Inlets. We have data that started in 2011 to present, and I'm currently working with Kim to try to get all that data into Matos and uploaded. One of the roadblocks that we've had, as I've heard many people mention in their presentations, is that resources. And so one of the issues we've had as a state agency is just having a lot of turnover in staff. So we've had a lot of hands on our data, and so we're just trying to work through and get it into the database. So if you do start seeing detections and pings coming from your data from previous studies and stuff, that's why, because we're finally getting some of this stuff in there. Our other array is the Central Southern Management Area. We started that in 2014, of course, and is still maintaining that now. And that's the Tar, Pamlico, Pungo, Trent, Neuse, and Cape Fear rivers. You can do it in the next slide, Kim. And we just wanted to kind of show just a general, this is kind of what the array currently looks like. We, as an agency, do not have any current acoustic projects ongoing. We do have a few holdovers from a previous striped bass acoustic tagging study that we did, but we try to maintain these because we know there's a lot of researchers out there with the universities and other partner agencies working on telemetry projects. So we're trying to maintain these. And we do that too also through our protected resources and making sure that we're tracking those, endangered species and threatened species. So we're got roughly 120 receivers out there, and we're maintaining those the best we can with the staff that we have. And again, just trying to get that data into Matos so you're able to access that. That's really short and sweet of what we're doing right now. Next slide, Kim. If you do have any questions, though, please feel free to reach out to me. This is my information, my direct phone number, and my email. I am happy to answer any questions that you may have and partner on anything that you may have questions about. Like I said, we've had a lot of staff turnover, so we're diligently working to get that data in there. And I'm hopeful by the end of the year, maybe we'll have everything caught up and good to go. So thanks for having me today, and great presentations, everyone.
Thanks, Amy. I think we will hold off on questions and we'll kind of get through all of these. And then maybe if there's time at the end, we'll do some time for questions, but feel free to, keep the conversation going in the chat. Next up is Margie from Virginia DWR, and she will be talking about some of her blue catfish work.
Hi. So I realized in hindsight, I probably tried to cram too much into this speed update, but that's my MO. So just a short background. We started this project in 2021. For anyone who's not familiar with blue catfish, they're an invasive species. They're a recreationally important species, and there's different commercial fisheries for them throughout the bay watersheds. DWR's goal in managing blue catfish is to facilitate increased removals to minimize the impact of these fish on native and commercially important species, but also to maintain a really economically important trophy fishery. So we have the goal in kind of starting this project was really to answer a lot of different information gaps and address a lot of questions that would facilitate better management and would facilitate better relationships with our anglers and encourage engagement with the fishery. So the first goals 1 and 2 are just really the standard, where are they and why? And then the third, we have a commercial low frequency electrofishing fishery for blue catfish. And we wanted to look at actively tracking these guys around the fishery to see how they're reacting and, if they were unavailable and how long, just to really increase the operational efficiency of commercial fisheries with the goal of distributing all of this information to our constituents to, again, facilitate removals and increase engagement with the fishery. So like I said, we started in 2021. We have 80 tagged fish out there. That seems like a lot, but because this is a commercial and recreational fishery, we're counting on losing quite a bit of fish, and they notoriously lose tags. So the retention is about 75% if everything goes perfectly. And it did not, because mine is around 75, but not too shabby. So I'll just kind of buzz through the rest of this. I do want to say that I'm kind of just sticking with the 2021 fish. There were some big changes to the James River array. So I'm kind of incorporating 2023 data, but having to kind of shift my approach a little bit. Go to next slide. So this is just a blast of your kind of standard abacus plot, colorful movement data representations that we're used to. So one of the really cool things, and so this is kind of getting at goals like 1 and 2, just some of our preliminary results from 2021 is we use network analysis to kind of look at how these community structure is throughout the river. One of the big concerns is these fish moving into lower reaches of the river, feeding on commercially important blue crab. So the population isn't well quantified, that overlap in range isn't well quantified, and so this really helped us. Is kind of one of the steps in looking at like what that overlap really is. And so we kind of saw the similar fallout of, movement tactics that you can see in migratory fish, where some moved a lot, some moved a little, and most were kind of in between. What was really interesting is we had, communities 1 and 2 really fell out to more heavily using upstream and lower and downstream areas, which was really interesting. So we're going to kind of dig into that a little bit more. Another interesting thing is, we often deal with how are we going to sample this population above and below the salt wedge. We rely on low frequency electrofishing, which doesn't work in highly, very salty water. And it's also temperature dependent. But, we saw, where we sample in July, August, that these fish are really restricted upstream where we do access them and sample. So that was really good to see too. On the right is just kind of how, a very messy diagram of how those communities kind of fell out. But really interesting. I won't get into methods, but I ran an optimal community algorithm about 1,000 times, and these communities were consistent membership across the board. So that was pretty interesting. But again, we'll see how this kind of plays out over time. You can go to last slide. So the really big takeaways, really surprising is we saw tributary fidelity. So we tagged a cohort in Powell Creek. Fish that were tagged in Powell Creek never went into any other tributary. Fish that we tagged in the main stem never went into any tributaries at all. And, we're thinking that the community structure kind of, is paired with maybe movement tactics, but then also possibly preferred prey. So again, low downstream habitat use, really consistent high density areas, which was really good to see because that's information we can again pass on to our anglers and our commercial license holders. On the right is just kind of a seasonal breakout of those communities and how those happened kind of in the first year. So really interesting for us. We're going to kind of keep this going. They're 10-year tags, but again, I doubt we'll get to that point, but hopefully I will have a fuller data set to show you guys results in the next meeting. But yeah, and if anybody wants to, I will say too that DWR, like this project has really been kind of proof of method for DWR to, show that we can answer a lot of big questions and, we can engage constituents and we can do cool stuff internally. And so, we are looking to expand the array in other tidal rivers. So if anybody is interested in collaborating and kind of, working together to kind of get funding and make that happen, feel free to reach out because I think that can only benefit a lot of our regional collaborators. So turn it back to you, Kim.
Thank you. So next up is Elizabeth from George Mason University talking about their arrays.
Hi, everyone. My name is Elizabeth Greenheck. I'm speaking on behalf of Reed Nelson, who could not be here today, about a couple projects we have coming up this year through 2026 in the Potomac River and Patuxent Rivers over here in the Chesapeake region. So the first, in terms of tagging, the first project we're going to be doing is tagging striped bass in the Patuxent River in collaboration with the Nature Conservancy and UMSES So that's going to be looking at some recreational post-release mortality. We're going to put out 100 tags in the spring and summer. These are going to be kind of short life tags, so about 60 days, and 100 are going to go out in total. So that's kind of one project we're doing. The other is going to be a bit more long-term, in collaboration with CERC and the Rappahannock tribe, tagging blueback herring and alewife. So blueback herring in 2024, alewife in 2025. We're going to be putting out some internal tags, so about 100 tags, specifically in the Potomac River. So 50 and 50 over the course of the next 2 years. And then next slide. And then in terms of arrays, we're going to be maintaining 2 arrays in the Patuxent and the Potomac rivers. So within the Patuxent River, we kind of have the array spread out across the river. So we've got 21 receivers that we're going to be deploying in the Patuxent from February 2024 to September 2024. So coming up pretty shortly. And those are just going to be out over a shorter period of time. In the Potomac, we're going to have a couple gates, one near the mouth of the Potomac River. So we're going to have 13 receivers, which we're hoping will give us 100% detection coverage, kind of in that lower portion of the Potomac. We're also going to put 7 receivers across the US 301 bridge, again, hoping to kind of gate that off and have 100% detection coverage. And then we're going to be deploying 6 receivers within Gunston Cove as well, which is kind of downstream from where we're going to be tagging these river herring in the Potomac. And it's a super short update. We don't have any data collected for us to report on, so probably more to come next year. But just a quick update for what's to come shortly.
Thank you. John from the Atlantic Shark Institute is going to be speaking next.
Yep, can you hear me?
Yes.
Super. All right, Kimball, thanks a lot, everybody. Appreciate the time. I'm just going to speak for a couple of minutes about this growing array that we have here in Rhode Island. It's really a collaboration between ourselves at the Atlantic Shark Institute and the Rhode Island Department of Environmental Management, who's been a great partner for us. I think the thing we're most excited about here in Rhode Island, at least we are, is the growth of this array. If you look at the screen, you can see the blue is the Rhode Island Department of Environmental Management receivers, and the red are from the Atlantic Shark Institute. And I think what's really interesting about that is if you look at the right-hand side of the column, you can see it wasn't a long, it was a pretty short time ago that there are actually no receivers here collecting data. And we had heard from a number of folks that there was a consideration that, and we actually got it in writing, people would talk about Rhode Island as being a little bit of a black hole, and we think we've solved a good chunk of that. What's interesting also is if you look at this, you can see the coverage is pretty consistent and pretty unique in the sense that that's us and that's Block Island, for those of you who are not that aware of Rhode Island and what it looks like. And up there where you see all those blue receivers from the Rhode Island Department of Environmental Management, that's Narragansett Bay. So we've got an awful lot of coverage. At this point, we've got 43 receivers, and we suspect that's going to grow this coming year. We have a number of studies, if you on our site and on the DEM site, that we're working on, over a dozen actually, that involve a number of species of sharks and other fish here in the state. And for us, we're actually going to be expanding left. That furthest island on the left there, that's Fisher's Island, New York. We got approval for 2 more studies from the state and from the local jurisdiction and the Coast Guard. So we're going to expand that way. And we're also going to add a couple of additional receivers at the tip of Block Island, the north tip, because we have a collaboration between ourselves and Rhode Island DEM and Mystic Aquarium and BIMI, Block Island Maritime Institute, Greg Scommel. So we're actually doing a shark seal interaction study, which is pretty fascinating. We were able to tag both white sharks, of which we've tagged a bunch at the Institute, as well as Greg, along with seals getting tagged by the aquarium and by others involved on that side of the equation, Rob DiGiovanni. So excited about the growth here and what's to come. Kim, if you want to go to the second slide. So far, here's what we're looking at. we've got over 3 million total detections since we started pushing this array out. Over 1,000 unique tags have been detected. And you can see that the species really varied, which is pretty interesting and pretty cool from our perspective when you look at everything from a loggerhead sea turtle to, blueback river herring to white sharks to, smooth hammerheads, et cetera. There's an awful lot going on here in the state of Rhode Island. And we think that the way that we've dispersed this array and the focuses of both organizations has really led to a lot of that. We've tagged approximately 100 sharks from the Institute's perspective, and that includes, acoustic tags and SPAT tags. And if you look through that, we're actually detecting a number of our own white sharks, and we've got makos and threshers and blues and poor beagles and sandbars and a whole host, black tip spinners that we have different projects on that we're pretty excited about. So our plan is probably to add 5 or 6 more receivers and continue to drive towards these projects. We've had a lot of people reach out to us excited about the fact that this is a newer array and they're getting a lot of data that they would not have gotten only a short time ago. So we're happy that we're helping others, and feel free to reach out if you have questions and have more details you'd like to know about what we're doing.
Thanks, John.
Thank you.
Next on the list is Ed from the New England Aquarium.
Yep. Can everybody hear me?
Yes.
All right. So hi, everyone. I'll start out with all the inshore work that we do here at the aquarium as part of the fisheries science team here. So we have an array set up in Nantucket Sound, which is bounded by Cape Cod to the north and Martha's Vineyard and Nantucket to the south. And the locations of all those receivers are those black dots on the map there. And those are largely determined by where the Coast Guard puts out aid to navigation buoys. So we'll talk to the Coast Guard about where we want to put them on the buoys. And those moorings consist of some stainless steel cable and a PVC sheath that will hose clamp the receivers to, add some sash weights at the bottom to make sure that they're not flapping around in the current too much. And then this past year, we've also put out some ARs on the bottom, and those have turned out pretty well for us in some high current areas. So we'll be continuing that this year as well. And then lastly, Caroline Kaletos, who's a PhD student in our lab, also maintains an array around Nantucket itself in collaboration with Greg Skomel from the Massachusetts Division of Marine Fisheries, as well as the Atlantic White Shark Conservancy. And so between those 3 setups, we have a pretty good coverage across Nantucket Sound, and that array is in there from about May through late October, early November each year. So we have 3 ongoing projects within this array. The first is Little Tunny, or as they're more commonly known, false albacore. So they're a pretty short-term visitor to Cape Cod and just the Northeast in general. They'll arrive in like late summer, stay through the fall, and they're targeted pretty heavily by catch and release recreational crowd during that time. But there's not much known about their movements either here or abroad. So we've been tagging them for the past 2 years and finding that they're making pretty extensive movements all the way down to Florida and then back based on detections we've been getting back from the ACT network as well as some other regional networks. So big thanks to those of you who've been contributing data there. And then on top of all that, we're also deploying acoustic tags to look at post-release survival with them as well. And then next up on the list, Caroline, as I mentioned, maintains that array around Nantucket, which is extremely convenient for both us and her because she also happens to live there. But she's been tagging sandbars for her dissertation work. That's her in the bottom right corner there with the shark, and she's looking at presence and movements of that species around Nantucket. And then last but not least for Nantucket Sound, Dr. Cara Dodge, who is our resident sea turtle expert, has been looking at leatherbacks as well as loggerheads. So for the leatherbacks, she's been tagging them externally. And then for the loggerheads, actually, she's been able to take advantage of cold stunned sea turtles that get trapped in Cape Cod Bay, which is just to the north of that map there. They come into our animal care facility for rehab, but she implanted them with internal acoustic tags, along with the help of some of our resident vets, monitored their health, and then eventually released them into the wild with those tags still on them. And that's actually the first instance of sea turtles being implanted with tags, and they published that work just a couple months ago. So definitely go check it out if that's something that might pique your interest. And then Lastly, we have one project a bit further to the north in Boston Harbor on sand tigers that's spearheaded by Dr. Ryan Noddick, who just started tagging them this past fall. That's continuing and expanding some previous work with telemetry on juveniles in that area. And he just deployed 8 more tags in the Boston Harbor Array that's maintained by the state. And he plans to continue that work this year as well. So next slide. And then really quickly here, we also maintain an array of 70+ AR receivers in offshore southern New England, where we work with the wind energy companies that own leases in those areas on the map there. So if you look at all of those dots, those are where we have our receivers. And if it looks a little bit haphazard, that's actually because we try to place the receivers in a way where they're not actively getting trawled up because there's a lot of mobile fishing gear there. And so we're placing them in areas where we know that there are things like hangs or wrecks. And then we've been tagging a whole bunch of species there to the right that are pelagics that are commonly seen and fished for in this area, as well as 2 bottom species, black sea bass and tautog. And we're looking at the effects of all that construction and then the entire installation of the wind turbine array that we'll have on the species in this area. And Jeff Beebone is the one that's currently heading up this project. There's some data sharing in place, but he's working on getting a data sharing agreement together for all of the wind companies. So that'll be forthcoming. And hopefully we'll be continuing this project on through 2025. So there'll be more tagging and monitoring taking place. And that's all we have.
Thanks, Ed.
Yep.
And our last one of the scheduled project updates is Bill from the Massachusetts DMS.
Hi, thank you, everyone. And thanks, Kim and Matt, for organizing another great meeting. So it's good to have these annually. So my name is Bill Hoffman. I work for Division of Marine Fisheries. For those who do not know, among a lot of other things, I am a part of a group that conducts a lot of acoustic telemetry up off Massachusetts. But today I'm going to be talking about a study that is not a DMF initiative, rather it's more of a NOAA initiative that we're a collaborator on. So Jim Hawks, who would typically be giving this talk and who is a collaborator on the study, and he was unavailable, so I'm going to step in for him. The name of the study is a cross-taxa assessment, habitat use and connectivity relative to marine protected areas in the Gulf of Maine, with the implications for management. And really the object of this study is to evaluate how highly migratory and protected species, including cetaceans, fish, pinnipeds, seabirds, turtles, are using a network of the state and federal MPAs within the Gulf of Maine and how their use interacts with humans. So I think the ultimate goal coming out of this will be advancing the science and understanding needed to protect coastal and ocean resources, and how a lot of these different animals are benefiting or using, or maybe not benefiting from the use of MPAs. So, so as you can see that it's a little bit different than what I usually do. Typically, I'd be more involved with like a very specific question, you know, answering a management question or a biological question. This is a little bit higher level, overarching, and so as you can imagine, there's a lot of different people involved with their expertise. And so the majority are through NOAA and this Fishery Science Center, but we also have collaborators with the Stellwagen Bank National Sanctuary, Marine Sanctuary, and then us, of course, and then Integrated Statistics, which is their contractors that work directly with the Science Center. Next slide, please. And so assessing the MPAs, specifically the Stellwagen Bank Sanctuary, located where that star is, Greg Scomo went through the ACT and came up with a map of where we had coverage throughout New England. This is a little bit dated, but it was pretty good, especially 2 years ago when we started the study. And one of the things that became apparent is that coverage within the Stellwagen Bank area was absent. So to address this, the group contracted commercial fishermen, specifically lobstermen, to deploy and manage the array for them. This is a little bit of a novel approach, But what they were able to do is they were able to capitalize off of another study that NOAA was conducting. So as everybody has heard by now, that at certain times of the year, Massachusetts, off the coast of Massachusetts, is the epicenter for right whales. And one of the biggest threats to right whales is fixed gears, specifically the vertical lines that support buoys going down to fixed gear. And so what they're developing right now and testing is essentially a pop-up buoy. So it'll be a buoy that is attached to the bottom to the lobster trawl, and then on demand, they press a button, acoustically it pops up to the surface. So when they do that, and part of the study is that every time they pop it up and haul their gear, they're getting location and time information. And then inside these lobster traps, they put in VR2s, I believe, VR2s. So, and then we told them, about where we want them. The gear's deployed, and then at the end of the year, we get the receivers back, and researchers will download the detection information. Next slide. So where we're at in this process, we're just getting towards the end of the field work. Both Greg and I will have tagged 4 species. Greg Skolmel and I've tagged about 70 animals apiece, plus Greg tagged a basking shark. I've tagged about 52 striped bass. And these were opportunistic too for me because they were also complementing other ongoing studies that I had going on. So for example, the striped bass was also part of a Circle Oak study. And the winter flounder, that was 70 fish, but we have 200 winter flounder out there right now as another study that we're doing. But it works perfectly for applying it to this and assessing winter flounder going into these into the marine protected areas, the MPAs. And so after this, starting in 2024, the individual researchers are going to be doing their preliminary analysis, doing their own work. And then it's anticipated that PhD students can be brought on board to merge these final results and conduct the final analysis. And so we just wanted to take this opportunity to just let you know this is a bigger project that's going out there. You will see co-op detection data being sent to you through our new array. But also is possible once they start the final analysis that someone will be reaching out to you asking about your tags. And obviously along the guidelines outlined by Mados, they may ask to some species ID information so they can document species that have been in and using the Stellwagen Bank Sanctuary. And that's what I got. Thank you.
Thanks, Bill. All right, we've got some extra time. Does anyone have any questions for the rapid fire update presenters?